Welcome to this video in which we will see about cytoskeleton, its structure, types and functions. As the name suggests, cytoskeleton is like a skeleton of the cell. As the skeleton supports the body, the cytoskeleton, which is made up of protein fibers, it supports the cell. Introduction to cytoskeleton. The cytoskeleton is a complex network of protein filaments that extends throughout the cytoplasm of eukaryotic cells. It acts as an internal framework that maintains the structural integrity of the cell, organizes the internal components of the cytoplasm. The cytoskeleton is responsible for maintaining the cell shape, providing mechanical strength, enabling cell movement, and facilitating intracellular transport of organelles and vesicles. Earlier, the cytoplasma was considered as a simple fluid in which the organelles are suspended. However, modern microscopic techniques such as fluorescence microscopy, electron microscopy have revealed that cytoplasma contains an organized network of protein filaments forming the cytoskeleton. One of the most important characteristics of the cytoskeleton is that it is highly dynamic. The protein filament continuously undergo assembly and disassembly depending upon the physiological requirement of the cell. This dynamic property allows cells to change shape, migrate, divide, and respond to environmental signals. The cytoskeleton also interacts with many cellular components such as membranes, organelles, and membrane motors. Through these interactions, it coordinates numerous cellular activities including intracellular trafficking, mechanical support, and signal transduction. Now we will see the basic concept of cytoskeleton. Cytoskeleton is composed of long protein filaments that forms an interconnected network throughout the cytoplasm. These filaments differ in size, composition, mechanical properties, and cellular functions. The cytoskeleton performs several important roles in the cell, such as maintains the shape of the cell, organization and positioning of the organelles, intracellular transport of vesicles and macromolecules, generation of mechanical forces for movements, participation in cell division, formation of specialized structures such as cilia, flagella, and microvilli. The cytoskeleton works together with motor proteins that converts chemical energy from ATP into mechanical movement. Three major filament systems that form the cytoskeleton includes microfilaments, intermediate filaments, and microtubules. Now we will see the types of cytoskeletal filaments in detail. The cytoskeleton consists of three major classes of protein filaments. First, microfilaments, second one is intermediate filaments, and third one is microtubules. If you see the diameter of these filaments, microtubule diameter is approximately 7 nanometer. Intermediate filaments, since their diameter is intermediate between microfilaments and microtubules, they are called as intermediate filaments. The diameter is 10 nanometer, and microtubules, the diameter is approximately 25 nanometer in size. The main proteins present in these particular filaments: actin in microfilaments, in intermediate filaments fibrous proteins, and in microtubules tubulins. Major functions of these filaments: microtubules cell movement and shape, intermediate filaments mechanical strength, and microtubules intracellular transport and cell division. The first one, cell movement and shape, is the main function of actin filaments. Each filament has a distinct molecular structure and performs specialized functions. Now we will see first details about microfilaments, which are made up of actin filaments. Structure of actin filaments. Microfilaments are the smallest cytoskeletal filaments with the diameter of approximately 7 nanometer. They are composed of protein actin, which is one of the most abundant protein in eukaryotic cells. Actin exists in two forms: G-actin, that is globular actin. This is a monomeric form of actin actin protein. Each G protein molecule binds ATP or ADP and has a molecular weight of approximately 42 kilodalton. F-actin, filamentous actin. When actin monomers polymerize, they form long helical filaments called F-actin. Polymerization of actin filaments. Actin polymerization occurs in three stages: nucleation, small actin oligomers are formed; elongation, additional actin monomers rapidly attach to the growing filaments; and the third step is steady state. Rate of addition equals to rate of dissociation of actin monomers. Actin filaments have structural polarity with two distinct ends: plus end where the faster growth takes place and minus end where the slow growth takes place. This polarity is important for directional movement of motor proteins. Actin filaments organization. Actin filaments can organize into different structures: parallel bundles, which are present in microvilli; contractile bundles, which are present in muscle fibers; and gel-like network, which is present in cell cortex. These arrangements are controlled by actin binding proteins such as fibrin, alpha-actinin, filamin, and spectrin. These proteins cross-link actin filaments into specific architecture. Location of actin filaments. Actin filaments are mainly located beneath the plasma membrane in the cell cortex, that is central part, in microvilli, in lamellipodia and filopodia, and in muscle cells. Functions of actin filaments: maintain cell shape. Actin forms a dense network beneath the plasma membrane that helps maintain the mechanical stability of the cell surface. Cell movement. Actin polymerization pushes the plasma membrane forward during the cell migration. Muscle contraction. In muscle cells, actin interact with myosin filaments to produce contraction. Cytokinesis. During cell division, actin forms a contractile ring that divides the cytoplasma into two daughter cells. Formation of cellular extensions. Actin supports structures such as microvilli, filopodia, and lamellipodia. Now we will see motor proteins associated with actin filaments. The primary motor protein associated with actin filament is myosin. Myosin structure. Myosin molecule consists of two heavy chains, two light chains, globular motor head that binds actin filament. The motor head hydrolyzes ATP to generate movement. Functions of myosin: muscle contraction, vesicular transport, cell migration, and cytokinesis. Myosin typically moves towards the plus end of the actin filaments. This was about the coming to the second type of cytoskeletal filaments, which are intermediate filaments. Now we will see the structure of intermediate filaments. Intermediate filaments are rope-like fibers with a diameter of about 10 nanometer. They are composed of family of fibrous protein that vary depending upon the cell type. The examples includes protein keratin, which is present in epithelial cells; vimentin, which is present in connective tissue cells; desmins, which are present in muscle cells; neurofilament, which is present in nerve cells; and lamins, which are present in nuclear envelope. Molecular assembly of intermediate filaments. Intermediate filaments assemble through several steps. Two monomers form a coil-coiled dimer. Two dimers associate to form a tetramer. Tetramers assemble into protofilaments, and protofilaments pack together to form a mature filament. Unlike actin filaments and microtubules, intermediate filaments lack polarity and do not bind ATP or GTP. Mechanical properties of intermediate filaments. Intermediate filaments are extremely strong and flexible. Their rope-like structures allow them to withstand mechanical stress. Location of intermediate filaments inside the cell. They are present in cytoplasm of many cells, epithelial tissue, muscle cells, nerve cells, nuclear lamina beneath the nuclear envelope. Functions of intermediate filaments. The first, as we had seen, it gives mechanical strength. They provide resistance against mechanical stress. Structural support. They help maintain the structural integrity of the tissue. nuclear structure, lamins from the nuclear lamina which support the nuclear envelope, cell junction stability, intermediate filaments anchor to desmosomes providing mechanical stability between adjacent cells. Now the third type of cytoskeletal filaments is microtubules. Structure of microtubules. Microtubules are the largest cytoskeletal filaments with a diameter of 25 nanometer. They are composed of protein tubulin, tubulin subunits. Two types of tubulin forms a heterodimer, alpha tubulin and beta tubulin. These dimers assemble into protofilaments. 13 protofilaments align side by side to form a hollow cylindrical tube-like structure. Microtubule polarity. Microtubules also possess polarity, plus end rapid growth and minus end where the slow growth takes place. Dynamic instability. Microtubules undergo process known as dynamic instability in which they rapidly switch between phases of growth and shrinkage. This behavior is regulated by GTP hydrolysis on beta tubulin. Microtubule organizing centers. Microtubules are nucleated at microtubule organizing centers, MTOCs. The most important MTOC is the centrosome, which contains two centrioles and pericentriolar material. Gamma tubulin complex helps initiate microtubule formation. Now we will see functions of microtubules. Microtubules play numerous roles in cellular organization, intracellular transport. Microtubules act as tracks for the movement of vesicles and organelles. Mitotic spindle formation. During cell division, microtubules form mitotic spindles which separates the chromosome or sister chromatid arms. Cilia and flagella. Microtubules form the structural framework of cilia and flagella. These structures contains 9+2 arrangement of microtubules which helps in movement of the cells. Maintenance of cell shape. Microtubules help maintain the elongated shape of cells such as neurons. Motor protein associated with microtubules. Two major motor proteins move along microtubules. Kinesins. Kinesin transports cargo towards the plus end of the microtubules. Functions include vesicle transport, movement of organelles and axonal transports. Dyneins moves toward the minus end. Functions include movement of cilia and flagella, intracellular transport and positioning of Golgi apparatus. Cytoskeleton and cell movements. Cell migration involves coordinated activity of cytoskeletal elements. Steps include actin polymerization at the leading edges, formation of cell adhesion, contraction of actin-myosin bundles, detachment of rare parts of the cells. This process allows cells to move through the tissue during development, immune responses and wound healing processes. Cytoskeleton in cell division. During mitosis, microtubules form mitotic spindles which separate duplicated chromosomes. Actin filaments form the contractile ring during cytokinesis during the cells, dividing the cells into two daughter cells. Importance of cytoskeleton. Cytoskeleton is essential for many cellular processes. Major functions includes maintenance of cell shape, organization of intracellular structures, intracellular transport, cell motility, cell division and mechanical support. Disruption in cytoskeletal proteins can lead to disease affecting muscles, nerves, epithelial tissue. Conclusion. The cytoskeleton is a dynamic and highly organized network of protein filaments that provide structural support and functional coordination in eukaryotic cells. It is composed of three major filament systems: microfilaments made up of actin, intermediate filaments and microtubules. Each type of filament has a unique molecular structure and performs specific role in maintaining cellular organization, enabling intracellular transport, facilitating cell movement and supporting the cell division. Together with motor proteins such as myosin, kinesin, dynein, the cytoskeleton act as a fundamental framework that supports many essential biological processes inside the cell. Thank you.